Okay, so get this. A healthcare worker is caught replacing painkillers
Yeah.
with something else, like tampering with them,
Yeah.
and then 2 patients test positive for HIV.
Oh, wow.
Yeah, talk about a hospital's worst nightmare, right?
Oh, yeah, for sure. Yeah.
It's actually a scenario from our deep dive source today, the OpenStax Microbiology textbook, which you can find a link to in the description.
Very interesting.
Yeah, and it really highlights the kind of situation that could become a public health crisis.
Absolutely. Yeah.
So in this scenario, the hospital's scrambling, and they end up testing over 1,300 patients who might have been exposed.
Wow.
Yeah, that's massive. But it also makes you think, how does HIV testing even work on that scale?
Right.
Like what's involved?
Yeah, a lot of people probably don't even realize what goes on behind the scenes, you know?
Exactly.
It's not just a simple finger prick.
Exactly. So let's break it down for our listener. First and foremost, why even bother testing everyone?
Right.
Wouldn't that be overkill?
Well, imagine you were one of those patients. Wouldn't you want to know as soon as possible?
Oh, absolutely.
Early detection is really key with HIV. It's not just about, finding out who has it, but it's about getting people the treatment that they need as soon as possible.
Right.
And that can really make a huge difference in how the disease progresses.
That makes sense.
Yeah.
Yeah, early intervention, peace of mind.
Absolutely.
It all adds up.
For sure.
Okay, so practically speaking, how do they even test that many people? What's the go-to test?
So the most common initial test is called an ELISA.
ELISA, okay.
ELISA.
Yeah.
And think of it like a first responder,
Okay.
It quickly checks for any signs of trouble. In this case, it's looking for antibodies.
Antibodies, okay, back up for a sec. Those are like our body's defense system, right?
Exactly. They're like your body's wanted posters.
Yeah.
They're specifically designed to target a certain invader.
Right.
So when you get infected with HIV, your immune system starts making antibodies that are unique to that virus.
Okay.
And the ELISA test can detect those wanted posters.
So it's not actually finding the virus itself.
That's right. It's what we call an indirect test.
Okay.
It's looking for your body's reaction to the virus, not the virus itself.
Interesting.
Like a detective looking for clues, not the actual culprit.
I like that analogy.
Yeah.
Okay, so what about accuracy?
Right.
If it's just looking for these antibodies, could it be wrong?
You're hitting on a really important point there. No test is perfect.
Right.
With ELISA, there is a chance of what we call false positives and false negatives.
So you could test positive and not have HIV, or the flip side, test negative when you actually do.
Yeah, that's scary.
Yeah, that's scary.
It is. And false negatives can happen if you test too soon after infection.
Okay.
There's a period called the seroconversion window.
Okay.
Basically, the time it takes for your body to produce enough of those antibody-wanted posters for the test to even detect.
Got it.
So timing is everything.
So what about those false positives? How do those happen?
Well, sometimes antibodies from other infections can be similar enough to HIV antibodies that they trigger a positive result. It's not super common, but it does happen.
And in a situation like this, with 1,300 people being tested, even a small percentage of false positives would mean quite a few people getting incorrect and very stressful results.
Exactly, and that's why confirmatory testing is so crucial.
Okay, so let's say someone tests positive on the ELISA. What's the next step?
That's when we bring in the Western blot test.
Okay.
It's like going from a first responder to a specialist.
Okay.
Much more detailed and specific.
So how does this specialist work?
Instead of just looking for antibodies, the Western blot actually identifies specific proteins from the HIV virus itself in the patient's blood.
Okay.
It's like piecing together a puzzle. If all the right pieces are there, it confirms the presence of the virus.
Sounds pretty foolproof, case closed.
Well, not always. Even the Western blot can have some ambiguity. Sometimes the results are indeterminate, meaning they can't definitively say yes or no.
Wait, seriously, you can't give a clear answer?
It happens more often than you might think. And because of that, and the fact that they're more expensive and time-consuming, they're not used for that initial screening.
Right.
But they are the gold standard for confirming a positive ELISA result.
So in our hospital case, they wouldn't start with the Western blot, but they'd use it to double-check any positives from those initial ELISAs.
Precisely.
Okay, so let's bring it back to that fictional case study.
Yeah.
Out of those 1,300 patients, 8 initially tested positive with the ELISA.
Wow.
That must have been terrifying for those people. What happened next?
Right. It highlights how stressful this kind of situation is for everyone involved.
Yeah.
Of those 8, 5 were given the Western blot, and thankfully they came back negative.
Oh.
Likely those were those false positives we were talking about.
Right.
But one person's Western blot confirmed they had HIV, and 2 others had indeterminate results.
So one confirmed case, 5 cleared and 2 stuck in limbo.
Right.
What happens to those patients?
Well, they don't just leave them hanging. Those 2 patients with indeterminate results would then undergo further testing, usually something called a PCR test, to get a more definitive answer.
PCR, that rings a bell. Isn't that what they used to test for COVID?
You got it. PCR stands for polymerase chain reaction, and it's incredibly sensitive. It can detect tiny amounts of viral genetic material, in this case HIV, in a blood sample.
So instead of looking for antibodies or proteins, this one's going straight to the source, the virus's DNA.
Exactly. It's the most direct way to determine if the virus is actually present or not. And in our case study, both of those patients with the indeterminate Western blot results ended up testing negative on the PCR, so no HIV for them.
That's a relief. So we have 1 confirmed case of HIV. Out of those initial 8, did they ever figure out if it was connected to the contaminated syringes?
Unfortunately, no. The tests can't tell you where the infection came from, just whether it's present or not. So it's impossible to say for sure whether that person contracted HIV from those syringes or from somewhere else entirely.
That's frustrating. But at least they were able to get a diagnosis and hopefully start treatment.
Yes, and the hospital did take responsibility and ensured that patient received immediate care.
That's good to hear. It reminds us that even with all the precautions and technology, there are real people at the heart of this story.
Absolutely. And it brings us to a bigger point. What is life like today for someone living with HIV? It's not the automatic death sentence it once was, right?
Yeah. I feel like we've come a long way, but I'm not totally up to date on current treatments and everything.
Well, you're right. We have come a long way. Thanks to some incredible advances in medicine, people with HIV can now live long and healthy lives with the right treatment.
What kind of treatment are we talking about specifically?
The cornerstone is something called antiretroviral therapy, or ART for short.
RT. Okay, break that down for me. How does it work?
Basically, it involves taking a combination of medications that work in different ways to suppress the replication of the HIV virus.
So not a cure, but it stops the virus from spreading and doing more damage.
Exactly. And by doing that, ART dramatically reduces the amount of HIV in the bloodstream. We call that the viral load.
And that's important because
lowering the viral load has tons of benefits. First, it helps protect the immune system by preventing the virus from destroying those crucial CD4 helper T cells we talked about earlier. Second, it massively reduces the risk of developing AIDS, which is the most advanced stage of HIV infection.
So goal number 1 is to keep that viral load as low as possible.
You got it. And the incredible thing is that when ART is taken consistently, it can suppress the viral load to undetectable levels.
Undetectable, does that mean the virus is gone entirely?
Not completely gone, but the amount of virus in the blood is so low that the standard tests can't pick it up.
Wow, that's huge. What does that mean practically for someone living with HIV?
Well, for one thing, it means they're much less likely to transmit the virus to others.
That's a game changer.
Absolutely. It also means their immune system is stronger and they're better able to fight off other infections. Essentially, someone with an undetectable viral load can live a long, healthy life without HIV having a huge impact on their day-to-day activities.
That's amazing. It really highlights the power of these treatments. But how do doctors make sure the treatment is working? I remember you mentioned flow cytometry earlier.
You've got a good memory, Flo. Cytometry is a vital tool for monitoring how well someone's immune system is doing by regularly checking the CD4 cell count. Doctors can see if the treatment is working as it should and adjust if needed.
So it's a constant feedback loop: test, treat, monitor, adjust.
That's a great way to put it. It's a lifelong journey of management. Both ART, it's a journey that can be filled with hope and possibility.
This is so much more optimistic than I think a lot of people realize. But even with these advancements, I imagine there are still challenges for people living with HIV.
Of course, stigma and discrimination unfortunately still exist, and that can take a huge toll on mental health and well-being. Plus, even with effective treatment, there can be side effects from the medications, and sometimes the virus develops resistance, which means needing to switch to a different drug regimen.
It's a good reminder that HIV is still a serious condition, and we can't forget about the human impact.
You're absolutely right. And thinking back to that hospital case, they had a real ethical dilemma on their hands.
Oh, definitely. It makes you think, when is large-scale testing the right call? Where do you draw the line? Yeah, it's a tough one. On the one hand, you want to catch these cases early on and prevent the spread, but on the other hand, you have to consider people's right to privacy.
Right.
And the potential for discrimination. Like, it's not an easy call.
It really isn't. Large-scale testing for infectious diseases can be a really powerful tool, but it's not without its downsides for sure.
So how do you weigh those competing interests? How do you decide when it's the right call?
There's no easy answer, but I think transparency is key.
Yeah.
People need to be informed about why the testing is being done, what the potential risks and benefits are, how their information is going to be protected, all of that.
So no sneaky testing without people's knowledge or consent.
Absolutely.
Right.
Not informed consent is essential.
No.
Everyone has the right to know what they're agreeing to and to opt out if they choose.
Makes sense. And what about those who do test positive, who has access to that information?
Well, confidentiality is really important. There need to be very strict protocols in place to make sure that those results are only shared with people who absolutely need to know, like the patient's healthcare provider.
It's a good reminder that medical testing, especially for something as sensitive as HIV, comes with a lot of ethical baggage. It's not just about the science.
You're so right. It's about people's lives, their well-being, their sense of safety.
And speaking of well-being, we've talked a lot about the advancements in treatment, but are those treatments available to everyone?
That's a huge issue. Unfortunately, access to healthcare isn't equal.
Yeah.
Things like socioeconomic status, where you live, even your race and ethnicity can create barriers to access.
So even if we nail the testing and get people diagnosed early, there's still a chance they might not be able to get the treatment they need.
That's the unfortunate reality, and it highlights a much bigger problem we need to address, those systemic barriers, and work towards a healthcare system that's truly equitable for everyone.
Couldn't agree more. This has been such an eye-opening deep dive. We've gone from a fictional case study to the nuts and bolts of HIV testing, to the realities of living with the virus, and now to these bigger ethical and societal questions.
It's all connected, isn't it?
Definitely. So as we wrap things up, what are some key takeaways you hope our listener will walk away with today?
Well, first, I hope they have a better understanding of how HIV testing works. The different tests involved, the potential for errors, the crucial role of confirmatory testing.
Knowledge is power, right?
Yeah.
The more we understand, the less fear and stigma there will be.
Exactly. And second, I hope they take away a sense of optimism about how far we've come in treating HIV. People with HIV can live long and healthy lives with proper care. That's something to celebrate.
It really is. And it's not just about the medications themselves, but about providing holistic support, fighting stigma, and advocating for equal access to care for everyone.
Well said. And finally, I hope this deep dive has encouraged everyone to think critically about the ethical considerations surrounding public health initiatives like large-scale testing.
Right.
It's a complex issue with no easy answers, but it's a conversation we need to keep having.
Absolutely. This deep dive has shown us that science ethics and human stories are all intertwined, and we can't address one without considering the others.
I couldn't agree more.
Thanks for joining us on this journey. We encourage you to continue learning about HIV and AIDS, support the organizations working on the front lines, and be an advocate for compassionate and equitable healthcare for everyone.